This experiment was designed to test the effect of light intensity on the photosynthesis rate of plants. Discs of deoxygenated leaves were filled with a bicarbonate solution and then placed in water. The time for the first 5 discs to rise was recorded. The hypothesis was that if the light intensity were to increase, the rate of photosynthesis would also increase because there would be a greater amount of light energy to power the reaction.
The results seem to follow what was thought to have happened. As the light intensity of the lamp decreased, the time taken for the leaf discs to float to the surface increased. This is proven when the height of the lamp was increased, which stands for light intensity decreasing, causing the time taken for the leaf discs to increase. The height of the lamp from the glass beaker containing the leaf discs at 9 centimeters had a ET50 of 5 minutes 48 seconds, 12 centimeters had a ET50 of 9 minutes 1 second, and 15 centimeters had a ET50 of 14 minutes 43 seconds. In the graph shown, the 15 centimeters line had a much higher slope compared to the other two lines, depicting that light intensity and time are indirectly proportional.
As hypothesized, as light intensity increases, then the rate of photosynthesis also increases. As the lamp got closer and closer to the flask with the leaves photosynthesize faster, which create a release of oxygen, which in turn causes the leaves to float up at a quicker rate. As the light was moved further out, it took a significantly longer time for the leaves to photosynthesize, which led to a slower rate of ascendance. This proves that as light intensity increases, the rate of photosynthesis increases. Also, this helps the release of oxygen for humans and the intake of carbon dioxide, which also helps the humans due to the inability of humans to breathe in too much CO2.
Trends. The distance of the lamp to the beaker directly affects the photosynthetic reactions inside of the leaf discs. When the lamp was only 9 inches from the beaker, the first leaf disc took less than a minute and a half to reach the top. The average time for the leaf discs took 6 minutes and 55 seconds when the lamp was 12 inches away from the beaker. The leaf disc took the longest amount of time to reach the top when the bulb was 15 inches away from the beaker. The average time was 13 minutes and 11 seconds. As the lamp is moved further away from the beaker, the light intensity decreases, making the photosynthetic reaction slower.
In nature, photosynthesis is powered by energy from sunlight. In the thylakoid membranes are photosystems with chlorophyll molecules. These pigment molecules absorb light energy and the excited electrons are then redirected to the reaction center. In the Z scheme, a chain of proteins uses the energy from photosystem 1, P700, to reduce NADP+ into NADPH, and the electron hole subsequently created is filled by electrons from photosystem 2, P680. All electrons come from the splitting of water into hydrogen ions and oxygen. As more energy is available, the photosystems use more and more electrons to process NADP+, and the water splitting enzyme works more to meet the electron demand. Therefore, with increased amounts of light energy, there is also an increase in oxygen level.
Possible errors can include damaged leaves, contaminated solutions, missed timings, and a malfunctioning light source. However, these errors could easily be avoided through careful execution and better materials. For example, the fluorescent light used that projected a greenish light could easily be replaced with a regular incandescent bulb that emits a wavelength of light that is easily absorbed. Photosynthesis can be applied to many fields such as biochemistry and biophysics. For example, in rapid electron transfer reactions, photosynthesis is often used to track the path of electrons instead of using an enzyme-substrate reaction because photosynthesis has a much faster reaction rate and only requires an extremely short light pulse in order to trigger a reaction. This photophosphorylation reaction can also be applied to plant-related genetic modifications and protein engineering.